This is for Biology 223 Genetics. I want to do, so I have a video on problem 31 from chapter 14, genetic mapping in eukaryotes. And I want to do it backwards, and backwards then relates to the problem 26 of this chapter, and is like the homework assignment. And also, I want you to pay attention to figure 10, that is relevant, and also figure B, which is in the problems. Those are relevant about why these numbers aren't always linear and as expected. So, chapter 31 has, problem 31 has this cross of Drosophila, and this is a traditional problem in genetics courses for many decades. You can look back and see them, and so if you're ever likely to see problems from long ago, talk to your parents or your grandparents, it may be like this. So let's look at some of the conventions first. So Dr. Cambrese may have done that. It has a cross of a female. The female is heterozygous at the loci that are interesting. So it's heterozygous at every locus of interest. There are 5. And the male then is, as tradition for a test cross, because it doesn't undergo meiosis, then it must be the female that's heterozygous, and the male is homozygous recessive, or where this the X chromosome, it could be hemizygous recessive. The convention of showing one line here as if it's one linkage group and showing them in physical order should be in physical order, but because the order is not known and the linkage is not known yet, this convention is violated. The way it's shown, it would be they're physically in alphabetical order and there's one linkage group. However, that's not true. So if we look at the answer, so there's a series of genotypes which are actually recorded as phenotypes and then interpreted as genotypes in genotypes of the gametes that are produced by the female meiosis. The end result of that problem, and I have a video for that, the end result is that actually, and you can just look in the book, this problem answer is shown in the book, that the actual genotype turns out to be, following a proper convention, B plus D, A plus C, and the reciprocal, because it was heterozygous, BD plus AC, and that's one linkage group of 4 loci of interest, and E as independently assorting, so presumably on different chromosomes or possibly on the same chromosome but very far apart. And then that cross would be against that would be the female, and the male then, which is being used to generate the gametes for the test cross, and to be all recessive, so that really what we have probably then is we have a female that is true breeding B plus D, A plus C, crossed with one of a reciprocal. This is F1. Now we're doing a test cross to see, because the idea is in order to map these loci, we need to have an organism that's heterozygous at each of these loci. And we'd want to be maintaining them in a true breeding state, because we cannot freeze Drosophila. So let's see what that means. So that is the proper way to show the genotypes of the cross, and then the map that we would show would be to have one linkage group like this, where B, D, A, C is the order, that's equivalent, there's no polarity here. C, A, D, B is the same, and E over here. And we're told in the answer that B and D are separated by 5.5 map units, D and A by 4.3 map units, A and C by 2.7 map units. Okay, so I'm going to put some notes here. Note 1, that's not a D. First thing we see is there are 5 heterozygous loci. 5 heterozygous loci, which means that we should expect that each of those loci, it can be functional plus or null, without the plus, right? So it can be plus or null at each of those 5, which means we will have 2 times 2 times 2 times 2 times 2 possible gametes, right? So there should be 2 to the 5th, so that's 32. We expect 32 possible gametes. 32 possible gametes are possible. However, also notice that there are 2 linkage groups. So 2 linkage groups. So we have, by independent assortment, we have from one linkage group, from each linkage group, let me say it that way, each group, we have a reciprocal pair. And so thus we have, for 2 groups, we expect to get reciprocal quadruplets. Thus, we expect that. So what's going to happen then? What's going to happen? I'm doing the problem backwards, starting with this map and knowing this is the cross, what gametes are predicted. So that's point 3. What's going to happen? Well, the most common thing that will happen will be parental. Parental, no crossing over. So what do we expect? We expect we will have B plus D, A plus C, E. That we don't have to notate that. And because E, this group, and E, the 4 loci and E independently assort, we will also have B, D, B plus D, A plus C, E plus. Those are not reciprocal except of E. And we will have the reciprocal pair D, B plus, B, D, let me cross that out, B, D plus A, C plus E, B, D plus A, C plus E plus. So we will have a reciprocal quadruplet of 4 for parental. Notice then we have at B, 2 plus, 2 null. At D, 2 plus, 2 null. A, 2 plus, 2 null. At C, 2 plus, 2 null. At E, 2 plus, 2 null. These will be equally frequent, and we know that frequency will be less than 100% total, right? And at least equal or greater to 50% total. And because this is out of 1,000, so of 1,000 progeny, you can imagine, having typed and looked at Drosophila in the lab, you can imagine what it's like to look at 1,000 on a microscope, and you can look at some of the problems or see this in old papers. People looked at many flies. So because there are 4 of them, we expect each of these gametes, each of these phenotypes. Remember, what we're seeing here are the phenotypes of the progeny. And the phenotypes of the progeny, because it was a test cross against a homozygous male recessive, the phenotypes seen here represent the genotypes of the gamete produced by the female. Keep that in mind. Sometimes you'll see the problem just a table of these phenotypes with numbers, and everything else is implicit. So we expect then that there will be less than 250, less than or equal to 250, and greater than 125 each, on average. So what do we expect here? We expect that all of these together are going to be represent somewhere, and we can see that the distance are such. We can also estimate, well, the distance between B and C In total, that's not much more than 10. So we expect this to be probably 90% or something like that. I'm just very roughly expect 90%. Okay, so that's going to be the most common group, and we will, no crossing over, expect that. And the next will be thinking about what's going to be the next most common. Let me put it that way. Next most common class. So let's think of this this way. We're going to have, I'm gonna end this video by this discussion before we go, that think of this. We have 4 loci in one group and one on the other. So we can have single crossing over here, single crossing over here, single crossing over here, right? Each, and this is independent assortment, and that's already taken care of in our calculations by the quadruplets, by the reciprocal quadruplets. So we expect to see a parental group of 4. Then we expect to have 1, 2, 3 groups of 3 times 1 x crossing over. Expect to see 3 of those. Most, middle, and least common. And then we would expect to have in each of those 4 quadruplets. And then we would expect double crossing over, double crossing over, double crossing over. So 3 classes of double crossing over. Didn't need the x there. Double crossing over. Also most, mid, and least. And we expect to have 1 triple crossing over, 1 times 3 crossing over. And if you add that together, you'll say 1, 2, 3, 4, 5, 6, 7, 8. 8 times each of those will be a group of 4. That's 32. That matches what we saw up here. So that will account for everything. I'm going to stop here, and in the next section I will deal with, I will calculate what the most common, most frequent recombinant class will be.